enzyme kinetics. It's fun, and I've heard that you guys have been eagerly anticipating it. Of course, as always, if you guys have any questions, please let me know. Please don't throw anything at me. I'm going to try to make this as painless as possible for all of us. I've added a lot of notes to the slides, just and they hopefully will make sense. Maybe not during the lecture, but after you go back and read it. And again, if you have any questions, let me know. So here are the learning objectives. I did want to give some general study tips to you guys. The test is approaching next week. I just wanted to provide you with this information so to direct your focus on what's important. So any of the slides that say clinical provider or clinical significance, that's just general information for you guys to provide context on the importance of the lecture material. As I said, you won't have to know a structure by its name, but you will have to know the components of all of the structure that we talked about. And that means you'll need to be able to identify the components with like the chemical components. So there was one in the lecture, was it Friday? That type of, it's gonna be, you may see a question that looks similar to that, maybe, I don't know. And so if you guys see a drug, a disease, or a specific enzyme mentioned, I recommend knowing that information only in the context of what's on the slide. You don't have to do any additional research. Again, if you have a question about the context of it on the slide, please let me know. And then if I don't go over the material, there are slides that should have been hidden. I don't know if they are for you guys. Cross those out, burn them, delete them, do not even look at them. You're going to be learning that information in another class probably. So are there any questions about how to study or what to study for the exams? So one more thing I want to mention is the exams in biochemistry they're kind of factoidy, so you're going to be presented with a question that is similar to how it's presented in the lecture material. So it's not going to be no trick questions. I'm not trying to trick you guys. There's no purpose in doing that. I want you guys to just understand the material and concepts in this class. Questions? Okay. So this will not be on the exam. This clinical significance, of course. So basically, we're going to be studying enzyme kinetics today. This is a very important topic in biochemistry. It's important because this information will help you guys interpret the lab testing results that deal with enzymes. For example, the AST, ALT test. And also, probably more importantly, you guys will be prescribing and learning about a lot of different drugs. And understanding how the drugs work will tell you will help you treat your patient. There's a number of drugs that the enzymes that metabolize drugs are affected by the food you eat and also other drugs. So when you guys are prescribing certain drugs, you need to know what compounds or foods or other drugs they're taking, because that's going to affect the drug metabolism. So you want to make sure you don't have too little of the drug in the body or too much, because of course if you have too little, then that's going to it's not going to reach its therapeutic effect. And if you have too much of the drug, you're going to see toxic effects. And so that's what enzyme kinetics is going to tell us about, like how the drug catalyzing the enzymes that break down the drugs, how they're working, and it'll give you insight on how to best treat your patients. So we know that enzymes are proteins. They lower the activation of energy of the enzyme catalyzed reaction. And in doing this, they increase the rate of reaction. So the speed of reaction is faster due to an enzyme. And the enzyme after the reaction has been completed, it is able to go on and catalyze that reaction again, so it's not consumed in the reaction. Enzymes contain a structure called the active site. That's a region on the enzyme where a substrate is what acts on the enzyme, where a substrate acts on the enzyme. Amino acid residues from that active site on the enzyme will react with functional groups on the substrate to catalyze a particular reaction. So there's some variety in the specificity of enzymes. You don't need to know this table at the bottom. This is just giving you examples. So enzymes can catalyze a specific type of reaction on a single substrate. That's absolute specificity. You also have enzymes that act on a specific bond type, and that's bond specificity, of course. And then enzymes act on a group of substrates. So when an enzyme catalyzes a reaction, when the enzyme and substrate come together, they form a complex called the enzyme-substrate complex. So that complex is simply when the enzyme and substrate come together. The important part of this complex is when the enzyme product complex occurs in this pathway. And what that is that is when the amino acid residues on the enzyme in the active site interact with the functional groups on the substrate to produce a product. So I say again, enzyme product complex is where the reaction is catalyzed and a product is produced. And here's an example of an enzyme substrate complex. We talked about lactose, which is the enzyme. Lactase is the enzyme. Lactose is the substrate. When lactose interacts with the enzyme, it forms the enzyme substrate complex. That's just when those two molecules come together. You see that water is being added to break the bond between the monosaccharides in lactose, and when that occurs, that produces the enzyme product complex. Then the monosaccharides are released from the enzyme, and then the enzyme is able to continue on to catalyze this reaction again. Any questions? So there are two common models for how enzymes work. You have the lock and key model. This is an old model that's not really, it's not really thought as being accurate, and that's where both the enzyme and the substrate are rigid, and they fit together very rigidly to catalyze the reaction. Now, the common model that is preferred is the induced fit model, and in this model, both the substrate and enzyme are dynamic and flexible, and they work together to configure in such a manner that the activation energy is lowered. And so their configuration is the correct geometrical shape that will cause the reaction to occur in a faster and more efficient manner.
I have a question.
Yes.
So whenever this is the slide before this is an example of a disaccharide, correct?
Yes.
So whenever you consume like a monosaccharide, it is able to be metabolized much faster because it doesn't go through this process?
It's a monosaccharide is broken down in the cell, and we're going to talk about that in the next block. That's what the body uses to attain energy in the form of ATP. So glucose is the main monosaccharide that our body uses. It's a completely different process.
And polysaccharides, I'm assuming, is...
So polysaccharides are broken down into monosaccharides eventually, but of course, because they're so long, it's going to be a stepwise process. And they're broken down into that monosaccharide to also help us get energy. And that pathway, one of the first pathways in the next block is glycolysis. You guys heard of glycolysis? Yeah. And we're going to talk about that next semester.
So I'm sorry, the polysaccharides wouldn't go into the enzyme substrate complex then?
Yes, they will. Like for example, cellulose is a polysaccharide. Cellulase is the enzyme, but it's not gonna fit like this. It's gonna be cellulase acting on the bonds individually because cellulose is such a large molecule. So the enzyme is not that big. The enzyme is actually small, so it can fit in between the two. It can fit in between the polysaccharide to break the bonds. Check on learning. What is the active site of the enzyme? A section of the enzyme. In the induced fit model, what happens to the shape of the substrate that binds? It adapts to the substrate. Any questions about that? So now we're going to talk about some different classifications of enzymes. So we're going to talk about 6 different classes. Typically enzymes end in ACE, and that tells you what it's acting on. We just saw an example of that. Lactase acts on lactose. Also, enzymes can be named with common names, are digestive enzymes, chymotrypsin, trypsin, and pepsin. That's another nomenclature used. And then enzymes are named based on the type of reaction that they catalyze, and we're going to talk about some examples of that. So for the next couple of slides, you guys don't have to know specific examples. There's a couple examples in each of them, unless the definition doesn't really help you. And we're going to see that right now. So oxidoreductase, it catalyzes reduction and oxidation reactions. Well, that really doesn't help me. So I'm going to use the example they have here. Alcohol dehydrogenase is the enzyme right here, and what it does is that it's removing 2 hydrogens, dehydrogenase, that's how it's got its name. And so it's using a coenzyme, and this is an example of oxidation. Transferase, we know transferase is transferring a group from one substrate to another. The example here is alanine transferase. It transfers the ammonium group from alanine to alpha ketoglutarate. We're going to talk about alpha ketoglutarate a lot in the next block. And basically the end result is producing another amino acid. So here's one amino acid, and if our body needs glutamate, this is the process that it goes, the transamination or transferase. Hydrolase, it's adding water to catalyze the reaction. And we know that the lactose or lactase is the hydrolase is adding water. Proteases add water, and so do lipase enzymes. And so this is a protein, it's adding water and the protease enzyme to break down the protein into eventually single amino acids. Then you have lyase, which catalyze the addition or removal of a group without using water. And the examples here are decarboxylase, that's removing carbon dioxide, deaminase, removing an amino group, dehydratase, removing water, and hydratase is adding water. So those names are telling you exactly what it's doing. Does that make sense? Yes.
pay for all those like breaking, those like peptides and saccharides?
No, there's the last example we're gonna talk about does require energy, but these do not. Isomerases is, that's just rearranging the molecules on a particular atom. And then we have ligase. Ligase is actually requires ATP to join two molecules together. So, any questions about the different types or groups, classes of enzymes? So I created this table just for ease of studying. I would just focus on the definitions. If I were you, you can learn the rest of it if you want to, but I don't think you guys will understand that. All right, so match these type of reactions with an enzyme. What converts a cis fatty acid to a trans fatty acid? Which one? Isomerase. What about removes two hydrogen atoms to form a double bond? Say again. Dehydrogenase. Combines two molecules to make a new compound. Synthetase. Adds an ammonium group. Does that make sense? Those are all easy peasy. So we talked about some factors that affect proteins, and so of course since enzymes are proteins, those factors will affect the function of enzymes as well. That's pH, temperature, and then also the concentration of the enzyme and the concentration of the particular substrate that the enzyme is acting on. So we know that proteins require optimal temperature, which is around our body temperature. If they exceed that temperature, then they're going to denature, and that's going to render the protein biologically inactive. Right? We talked about that in a previous lecture. We know that enzymes, I believe I mentioned that enzymes in the digestive tract, for example, the stomach, the pH is low, pepsin works in the stomach, and then in the small intestines, trypsin acts there, the pH is between 7 and 8. So those enzymes cannot act in locations where the pH is drastically different. They're going to be rendered non-biologically active. So the pH environment of enzyme is specific to the specific type of enzyme. And then the substrate concentration. So as at a constant substrate concentration, if we increase the enzyme concentration, that's gonna increase the rate of reaction because there's more enzyme that the substrate can interact with. And at a constant enzyme concentration, when we increase the substrate concentration, the reaction rate will increase, but only to a certain point. And that point is maximum activity, shown here. Maximum activity is the point at which all the enzymes are occupied with substrate, or they're saturated with substrate. So, because we don't have any more enzymes available, the reaction rate, that's kind of the maximum rate of reaction. This concept is very important in the Michaelis-Menten equation that you guys are so eager and ready for me to talk about. We're getting there, so hang on, hold on to your seats. But yeah, that is a very important, saturation is a very important concept in the Michaelis-Menten equation. And so enzymes are regulated by a number of factors, either feedback, control, or positive or negative allosteric regulation. And so if we have enough of a certain product, an enzyme will stop producing that product. And if we don't have enough of a product, that will cause an increase in enzyme activity. For example, allosteric enzymes work by either positive or negative regulation. Allosteric enzymes have a site that is different from the active site where a regulator molecule will interact. When the regulator molecule interacts, it changes the conformation of the active site, making it, that active site conformation, either more favorable for substrate to bind, to produce a particular product, or less favorable because we have enough of that product. And so those are examples of positive and negative allosteric regulation. So in positive allosteric regulation, an allosteric regulator will bind, cause a conformational change of that active site, allowing the substrate to fit more effectively to result in a particular product. Conversely, for negative allosteric regulation, an allosteric molecule will bind to the enzyme, change the shape of that active site so the substrate fleshy cannot interact because we already have enough of a specific product. Does that make sense? So, inhibitors of enzymes basically stop the functioning of enzymes, and there are two types of inhibitors, reversible or irreversible inhibitors, and we're going to talk about both of those types. So, a reversible inhibitor, there's two subtypes of those. You have competitive, that competes at the active site. Non-competitive inhibitors compete at a site other than the active site. So we're gonna go over some slides. This slide is not testable. This is just providing context to a competitive inhibitor. And so that competitive inhibitor is Viagra. I'm just showing you the mechanism of action, and hopefully this helps in understanding how competitive inhibition works. So what happens is Viagra inhibits phosphodiesterase 5 enzymes. That enzyme is responsible under normal conditions for breaking down cyclic GMP. So what happens during sexual arousal, nitrous oxide is produced, which causes the conversion of GTP to cyclic GMP. That cyclic GMP causes increased arterial flow and an erection in men. So cyclic GMP is broken down by phosphodiesterase 5, and when it's broken down, the erection is lost. So what Viagra does is it competitively blocks phosphodiesterase 5 to increase the amount of cyclic GMP in the cell, which prolongs the erection. Does that make sense? So, Viagra and Levitra are competitive inhibitors. Competitive inhibitors have a structure that is similar to the natural endogenous molecule, in this case, which is cyclic GMP. So cyclic GMP is the natural endogenous molecule for phosphodiesterase 5 enzymes, and Viagra and Levitra are structurally similar to that. So what Viagra does is, if you see, here's the enzyme, and this substrate would be cyclic GMP under normal conditions. When the substrate interacts with the phosphodiesterase 5 enzyme, it basically inhibits arterial blood flow. That's what it's doing. That's the product, sort of. So, but what Viagra does is it competes. Viagra is this eye. It competes with the endogenous substrate And once it binds, it will stop the breakdown of cyclic GMP. And what that does is it prolongs the arterial blood flow and prolongs the erection. Does that make sense? Here's an example of a non-competitive inhibitor, cyanide. Cyanide acts on, this is the electron transport chain. So the electron transport chain is the final step in energy production. Electron transport chain, electrons and oxygen are passed along the different complexes. We're going to talk about this in much greater detail in the next slide. But electrons are passed along the complexes, and the end result is the production of energy in the form of ATP. So here's complex 4 where cytochrome C oxidase lives. Cyanide binds to an allosteric site on cytochrome C oxidase, and so what that does is that electrons still can bind, but the reaction is going to stop because the conformation is changed. And so that is the takeaway message. So in this example, non-competitive inhibitor, the endogenous substrate is electrons, the inhibitor is cyanide. So for non-competitive inhibitors, the structure of the inhibitor is not related to that of the endogenous molecule because it acts on a different site. What happens is this inhibitor acts on a different site and it changes the shape of the active site. So the normal reaction that is supposed to take place is stopped. Does that make sense? Any questions about that?
So it's still inhibiting, it's just not using the actual, like the active site to do so?
Correct. Yeah. So, and this pictorial diagram is just showing the same thing. So, this would be the cytochrome C oxidase enzyme. Electrons would be the substrate. Under normal conditions, the substrate binds, passes, and is able to produce ATP. That would be the product. Yes.
So a non-competitor or a non-competitive inhibitor is non-reversible because so non-competitive inhibitor is not reversible because it's using the allosteric site in the back door. So regardless of how much you increase the concentration of the substrate, it's never going to be able to fit the active site.
Right. So that's partially correct. It is reversible, but what happens is that that non-competitive inhibitor will either have to be removed from the allosteric site, and that is done by it just releasing after it's done what it needs to do. Or, like for example, in severe cases, dialysis is used to remove it from the system. Does that make sense?
Yeah. So basically, without taking until it runs its course, or without taking some kind of actual intervention, it cannot be displaced.
Correct.
Because it's using the allosteric.
Right. It's still no amount of substrate is going to continue that reaction because the active site is messed up, so the enzyme catalyzed reaction cannot occur. Yes.
Just wondering, how would adding more substrate reverse the effect of all the active sites for
Because it's like kind of, I have an analogy, musical chairs is the analogy I use in the slide, but it's kind of like you're competing with the overload of the competitive inhibitor is competing with the natural substrate. So that reaction is like, they don't stay on there forever. So once the natural substrate goes off, then the inhibitor comes on, and if there's more inhibitor, once the substrate comes off, the inhibitor is just gonna like flood the enzyme system. Does that make sense? Was there another question?
Yes, sorry. So in the case of this example specifically with cyanide being potentially reversed by hydroxycobalamin, or sodium vegetable vinegar. Can you give an example of how that works? Because
And thank you. So what happens is, the cyanide molecules bind with the hydroxycobalamin, sodium nitrate, sodium sulfate, and that will kind of like, they chelate to that molecule, and then that molecule is rendered non-toxic, and then we excrete it in our urine.
But so do they pull from the allosteric side on the end?
No.
Or do
They just have to be, all of the cyanide floating around, and when it decides to come off the allosteric site, that hydroxycobalamin is there waiting to form a covalent bond with it, and then that bond makes the cyanide non-toxic, and then it's able to be excreted in our urine. Does that make sense how this drug works?
So you're still waiting for it to definitely come off of the.
Yeah, and the if you have like a toxic amount, there's gonna be a lot floating around, so you'll give the hydroxycobalamin, and all of the cyanide floating around will be removed. And then once it's released from the enzyme, then it will bind. Does that answer your question?
Yes, thank you.
And so irreversible inhibition, this occurs when the inhibitor forms a covalent bond with the enzyme near the active site. So when that covalent bond is formed, basically you can just say the enzyme is dead, and our body's going to need to regenerate more enzyme via the translation process. And an example of this is aspirin. Aspirin covalently binds to COX-1, and when it does, that enzyme, it has to be degraded, and our body needs to build new, make new toxic enzymes. Okay. So, identify each description or inhibitor as competitive or non-competitive. Increasing substrate reverses inhibition. Competitive. It binds to the enzyme surface but not the active site. Non-competitive. Its structure is similar to the substrate. Competitive. And inhibition is not reversed by adding more substrate. Does that make sense? Any questions? Okay. And here's just a little table giving the summary of differences between competitive, non-competitive, and irreversible that we talked about. We have now reached enzyme kinetics. Yay! Right? Woo! Should I do a dance? No, because I haven't talked about this. So enzyme kinetics is basically studies how fast the enzyme works. There are many factors involved in enzyme kinetics. It's how much substrate we have, how much enzyme we have, the affinity of an enzyme to its substrate, and whether or not competitive or non-competitive inhibition occurs. And so I talked about this at the beginning of the lecture, but enzyme kinetics is very important in drug metabolism, and pharmaceutical scientists determine are the scientists responsible for determining the exact values for known enzymes and known drugs, and that is used to help determine how much drugs are recommended to prescribe. So there are two commonly used models that help us understand enzyme kinetics. They are the Michaelis-Menten and the Lyman-Burke models. You don't need to know the slide, this is just to provide context on the history. So the Michaelis-Menten model or equation of kinetics was developed to tell us how enzymes catalyze reaction and that relationship between the substrate concentration and how fast the reaction will occur. This model uses velocity V, which is the rate of the reaction, and it also looks at the substrate concentration, S, in addition to introducing the Michaelis constant, which is Km, and then V max is the maximum rate of reaction. So V max is when an enzyme is saturated and all of the enzymes are occupied with substrates. So us understanding the Km and V max of a particular drug and or protein helps us understand how efficient a reaction is. So the Lange-Burke model is the linearization of the Michaelis-Menten, and that takes the Michaelis-Menten equation and derives it into a straight line, and that allows us to better estimate the parameters, the Km and Vmax of a particular enzyme. We're gonna see plenty of examples of this. And so here's the Michaelis-Menten equation. V equals Vmax times the substrate concentration over the Km plus the substrate concentration. As I mentioned, V null is the velocity or speed of the reaction. Vmax is the maximum rate achieved by enzyme catalyzed reaction. That's when enzymes are fully saturated. So I forgot to mention, for these models that we're going to talk about, what's important for you guys to understand is all of the parameters. So what the parameters of the equations are, and then how to read them on the graph. And please let me know if you have questions once we get to the graph. And so a general rule that helps us, like a general cheat sheet rule that helps us know whether or not we're looking at competitive versus non-competitive inhibition. During non-competitive inhibition, there is no change in Km, but the Vmax decreases. In competitive inhibition, there's no change in the Vmax, but the Km increases. So here's the Michaelis-Menten graph for competitive inhibitor, Parameters, x-axis is the substrate concentration, y-axis is the speed of the reaction or the velocity. This green horizontal line is the Vmax, that means the enzymes are saturated. The red line is the endogenous substrate with no inhibitor. The blue line is the endogenous substrate with inhibitor. So if we take the example that we talked about earlier, the enzyme would be phosphodiesterase 5. The endogenous substrate is cyclic GMP, and the inhibitor is Viagra. And here are the Km values. The Km values it's a Michaelis constant that has been predetermined for known proteins, known enzymes and drugs. Km value is basically the substrate concentration at half of the max velocity. So basically the Km values are kind of values that help us know how much substrate we're looking at with the natural substrate versus the inhibitor. So the Km values are on the x-axis, and that pretty much tells us how well our enzyme and substrates work together. Are there any questions so far?
I got a question.
Yes.
So in an example of Viagra, if it's not working, is it because it's being metabolized too fast or not being metabolized?
Possibly, yeah. Possibly it would be it's metabolized too fast. Yeah. And we all have different ranges of how fast our based on our genetics, our health, like enzymes metabolize, there's different types. There's normal metabolizers, slow metabolizers, fast metabolizers. That could be one of the reasons that it's being metabolized too fast. So it's not enough in the system in order to produce the intended effect. So in this graphic equation, we see that the Km is shifted to the right for when we look at the blue line, which is the cyclic GMP and Viagra, right? So, and we also see that the Vmax, or the rate of reaction, the maximum activity of the reaction, it occurs really quickly without the inhibitor, but it takes a little while longer, but it does, with inhibitor does still reach the Vmax. So what this example is telling us is that if we add more cyclic GMP, the natural endogenous, with the inhibitor, if our body produces more cyclic GMP, it is still able to overcome the effects of the drug because the Vmax is reached. However, we need to add more of it, of course, so that is going to increase the substrate concentration. So as substrate concentration increases, competitive inhibition, there's no change in Vmax. We're still able to reach Vmax. Does that make sense? Any questions? So for the exam, for this, if you were to see a question like this on the exam, you would need to be able to identify what is going on. Is the Km the same? Is it different? Is Vmax the same? Is it different? And so you see Km is different very clearly, right? Yes, no, maybe, yes. You see the Vmax is the same. It takes a little longer, but it's still the same. It approaches that the natural in substrate alone. Does that make sense? Yes.
So you'd say like for this one, the Vmax.
Can you speak up a little?
Sorry,
No.
so you say for this one, the Vmax is constant, for Km is decreasing.
Km, you said increasing? Km is increasing, because this is substrate concentration increasing more in this direction. So this is the natural substrate, this is natural substrate inhibitor. So you need to add more natural substrate to counteract the effects of with the inhibitor. So Km is increasing, and it's still able to reach the Vmax. And so just to show you guys over here what I was saying, that Km, this line represents natural substrate. This is natural substrate with the inhibitor. You see there's an increase in substrate concentration of the natural agonist, the natural substrate. And so with the increase in the substrate, you're able to counteract the effects of the inhibitor to still reach Vmax. Does that make sense? Yes.
Yeah,
Mike?
initial confusion for me was Km greater than Km2 based off of like mathematical, but it's just that not mathematical symbol, it's just showing it increases to the right. Like if you read it mathematically, you'd read it.
Oh shoot,
Like
I put the
Km1 is
wrong.
greater than.
Yeah, sorry, thank you. I put that Km2 is greater than Km1, my bad.
Okay.
Yes. Sorry for that, I'm gonna fix that. Any other questions? Yes.
Can you ask me a question about this graph?
Yes.
Let me ask you about the next graph.
Yes, sir. I'll be here.
Yes.
So, since the oopsies that.
Can you speak up?
So since there's the oopsie with the greater than less than symbol, that.
I'm sorry, I can't.
My apologies, so since there was the typo with the greater than less than symbol, is the orange line then still no inhibitor?
Yes, it's just, this is just wrong, my apologies. KM2 is greater than KM1. I'm sorry, guys, I worked on this forever and I thought it was right, but still correct. Yes.
Okay, so I think I'm having confusion with the graph because my question is, why would I take the inhibitor if I'm gonna get there to the Vmax at the same time? Does that make sense?
No, say that again.
Why would I take the inhibitor if essentially like where your graph ends, they all meet at the same time? So what's the point of taking the inhibitor versus the natural substrate?
So, yeah, you want your erection time to be prolonged. So the inhibitor is a drug that's doing something that the body needs. So that's why you would take the inhibitor. This is just demonstrating that the natural substrate can overcome the effects of the inhibitor, and that tells you it's competitive inhibition.
Okay.
So the inhibitor is going to usually be some type of drug that our body needs or wants. It's confusing because why would you take it? But you would take it in this case because it's doing something that we want our body to do. Does that make sense?
So I mean this as serious as I can. So erection time is on the left side of the orange and blue line. So there's more erection time on the blue line, right? Is that the curve we're looking at?
Yes.
So orange line is obviously, yeah, so less erection time without the inhibitor.
Yes, sorry, yes.
Yeah, so I was looking at all at the left side.
Yes.
Okay.
Yes.
So, but if, with no inhibitor and with the inhibitor on this example, it's working on both of them, right? Like you don't need.
This is just what people did when they didn't have TV, social media, all that stuff to figure out how enzymes worked. Any other questions? So we already see that this looks different, right? So and this is an example of non-competitive inhibition. The parameters for the graph are still the same: substrate concentration, velocity on the y-axis. And if we take the cyanide example, the enzyme would be cytochrome C oxidase. We see Vmax. This is the electrons doing what they need to do to produce ATP. Cyanide acts on a different site and it distorts the shape of the active site where electrons act. So it's gonna completely drop the rate of reaction. It's gonna kind of stop the reaction from occurring because the electrons, even though some of them might still be bound, that conformational change is gonna cause the reaction to not be able to be completed. So what you see in this case is you see that a drop in Vmax. And again, because the electron concentration, it can be the same, and it can still bind, but it's still not gonna, the reaction is still not gonna occur because the inhibitor has distorted the active site. Does that make sense? And you guys can think about it on your 10-minute break and then come back. Right.
prolonged direction until you get to VO2 max. This is hard. So, in a competitive gym, you take a inhibitor, it's a close this is bad because remember this is whatever this is. You want to get to VO2 max. The side is you can't think of this.
It might be. When that.
Because on this one,
It's supposed to be half, but on mine,
so
when that graph is.
that's a bad thing. This is a good thing.
Like supposed to be half. Yeah. Yeah.
So that is a good thing.
Yeah, I would.
Yes, but then you want to get to the point.
Honestly, okay, yeah.
This is where I can just so that's probably.
It says it on the bottle, right? It's like 6 hours.
Yeah.
Look, down goes from 0 to 5, then 5 to 10, and down goes from 0 to 10 to 20. And then like they both end at 47. So you would just know that but they're one grade first. And then which one you want to do is a higher level.
I guess that's an example. This will probably one week. But enough
So it's saying KM is constant. Because they're both
Yes, that's the first. Yeah, this one.
I think it just like took off his
so that's like they follow each other at the same pace.
Yeah, so it's the feedback of this. This is happening. Yeah. Yes, yes.
Your Vmax.
So if you look at the x-axis,
Basically looking at this thing, is substrate versus K Vmax. Maximum rate achieved by the system. This is the most that you can find. So there's no possible solution to that. Like this is the extra right now.
it's gonna look
So that's the answer here.
This is completely different. We'll just change the colors.
the line basically and they're the same. So half this is
Where do we take a wrong turn? Where do we go from this? Actually, that's I feel like this is starting to make sense, and then we did this. I don't know how you guys are.
no, yes, I could have drawn it like that. But I could change it to where this can't. Yes, that's probably. Right here. Yes. I'll fix. I just pulled from the side and made it down here. Yes, but this, yes, I was waiting for somebody to it's, yeah, that is this is like okay, eventually.
Yeah.
You know, like what I said, that you know, get some stride.
And is that all we need to do? We don't need to do
Long term. Next man. A couple other times. Or you just use up-to-date. Like the like somebody's 35
Yeah, this is wrong. Because this came to is up here.
Thank you.
All right, guys, all right. Now we moving on to non-competitive inhibition. But y'all, does this make sense? Any questions about non-competitive inhibition? I was telling one of your classmates, you guys are gonna have to draw this for the exam. Now, you need to be able to identify the components, KM, listen up guys, you need to be able to identify the components, KM, Vmax, whether or not they're changing. And so being able to identify the different graphs, you should be able to do that just visually. And you need to understand what KM means, what Vmax means. Yeah, that's pretty much it. Yes.
Okay, one more time. When you identify where KM 1 and 2 is.
So KM is half of the Vmax. So half of 40 is 20. Half of 20 is 10. So that KM is the substrate concentration at half of the Vmax. Does that make sense? Yes.
So each line has a specific Vmax? That's how you just answer?
For non-competitive inhibition, because this is the Vmax is this, the Vmax meaning the rate of reaction has dropped for non-competitive inhibition. And why is that? Why has the Vmax dropped for non-competitive inhibition? Yes. Say again?
There's less functional enzymes.
Right, correct. And why are there less functional enzymes? Because the inhibitor binds to an allosteric site and it changes the shape of the active site. So that's gonna just stop the reaction from occurring. Does that make sense?
So on competitive, they have the same Vmax.
Right, so what does that mean?
I guess my confusion is KM2 is supposed to be greater than KM1, but they have the same Vmax. So how is that?
What is KM stand? What is KM looking at?
Half of the Vmax.
Right, that KM is half of the Vmax, but what value is KM giving us on this graph? The substrate concentration. So KM is increased for competitive inhibition, which then allows for the added substrate to compete with the inhibitor for the active site. And so more substrate binds to the active site, which allows it to reach Vmax still. Does that make sense? Yes.
I have kind of two questions. Will KM1 and KM2 always be clearly labeled like that? Like, could you switch those and then just change the equation? And then also, like, why do we use half of the Vmax as that reference point for the equation?
That's a good question. That's kind of, it annoyed me when I learned this stuff, like before you guys were born. That it's just a way, that is kind of a quick way to assess how much substrate concentration. So it's a constant that the scientists Michaelis and Minton developed that as a quick way to look at the graph and kind of get a value on if you add more substrate, will you still reach Vmax? So it's kind of like easier way to estimate what's going on with the substrate and enzymes in terms of affinity. Any other questions? All right, now Lineweaver and Burke. Lineweaver Burke is the double reciprocal plot. Basically, you see on your left-hand side, the Michaelis-Menten equation is derived into a straight line. The Lineweaver-Burke is a straight line plot where the y-intercept represents 1 over V max, the x-intercept represents 1 over Km. What is V max again? One person say it. I can't, because I can't hear. Yes. Maximum velocity of the enzyme reaction. Okay. So for competitive inhibition, x-axis substrate concentration, y-axis is 1 over V. The y-intercept is 1 over V max. The x-intercept is 1 over K, minus 1 over K. The red line represents no inhibitor. The blue line represents inhibitor. So what does it mean that the y-intercept is the same for both of these? Same V max. Does that make sense? And on the x-axis, you see this red line, no inhibitor, blue line, inhibitor. So that's right shifted, right? So that means a greater Km for this blue line. Does that make sense? So more substrate is needed in order to achieve the same V max. Any questions about this? Non-competitive inhibition looks a little bit different, of course. Same parameters. Red line, no inhibitor. Blue line, inhibitor. What do we notice immediately that's the same in this graph? And what is that? Km is the same, so what does that mean? Someone, anyone? One person say it loudly, please. Substrate concentration is the same. But we see that the y-intercept is different, right? So that means the V max is different. So if we're just looking at this graph, just like in our numerical minds, it looks like this blue line with the inhibitor is greater, since it's higher on the y-axis than the no inhibitor. But we know that Vmax drops in non-competitive inhibition, right? So what you guys need to keep in mind is that this is 1 over Vmax. So if no inhibitor is 1 over 5 and this inhibitor line is 1 over 10, of course 0.2 is greater than 0.1. So it's kind of the inverse. So this line being up means that it's actually reduced like we saw in this graph. Does that make sense?
Meaning that more time is required in order to reach Vmax.
You can't reach Vmax in non-competitive inhibition.
Right, but
So, are there any questions about Michaelis-Menten online with Burke? Again, you guys, if you have questions, you can please feel free to email me, see me after class. I'm here to answer any questions you have. And so this is not testable, but I just wanted you guys to, and I mentioned it, I believe, how are Km and Vmax determined? They are determined in a pharmaceutical setting. You guys don't have to worry about this. And so databases already have these values predetermined for known drugs and enzymes. And so we talked about Viagra. Viagra is metabolized at 384 primarily, and so the KM for Viagra is 221, Vmax is 65. So what the KM and Vmax is telling us about Viagra is that Viagra competes sufficiently with the endogenous substrate. However, it takes a long time for Viagra, or a longer time for Viagra to clear the system, so it may present with some side effects. Are there any questions? So here's the musical chair analogy. Hopefully this helps. I thought about this and I hope it helps you guys understand it. So in the musical chair analogy, the seat is the active site, the arms are the allosteric site. The adult would be the substrate and the child is the inhibitor. So in competitive inhibition, the adults and children are competing for that seat, the active site. If we add more adults, the KM will increase and the adults will probably occupy all of the seats and they will reach the Vmax and win the game. Does that make sense? Does this analogy make any type of sense? Please, somebody say yes. Because I thought about this for a long time. Thank you. Non-competitive inhibition, the seat is still the active site. The arm, even though there's no arms on these chairs, are the allosteric site. So the kids will sit on the arm, and that will probably make the adults not want to sit on the seat. So what that's going to do is reduce the adults occupying the seat because the children are sitting on the arms, and so that's going to slow down the reaction. So with the equal number of adults and children, if the children sit on the arm, the adults are not going to sit on the seat, and the game is going to be kind of over because the kids didn't play correctly. Does that make sense? Okay. All right. So these are just some little takeaways from McKay-Smithson. What does a low KM tell you about how the enzyme binds to the substrate? A low KM means that less substrate is needed to interact with the enzyme. So that means that low KM indicates a high affinity of the enzyme to the substrate. And so what happens to KM and Vmax if a competitive inhibitor is present? The competitive inhibitor fights with the substrate for the active site, the musical chairs. And so KM increases and Vmax stays the same, meaning that if you add enough substrate, you can still reach the top speed. All right? Now we're shifting gears to talking about coenzymes and cofactors. So we talked about macronutrients, proteins, lipids, and carbohydrates. Now we're going to talk about micronutrients, which are cofactors and coenzymes. Those are very important for catalyzing reactions. They're involved as supporting cast members of catalyzing reactions, and we're going to talk about different examples of those. But before we do, we're going to talk about zymogens, which are proenzymes. So I would recommend you guys knowing the zymogen in its inactive state and the active state. And so zymogens are very important. We've talked about several of these, the ones involved in the digestive tract, chymotrypsin, pepsinogen, and trypsinogen. And then we're going to talk about fibrinogen and prothrombin when we talk about blood clotting. So typically zymogens, once they are activated, they either require a cofactor to be activated or they work with coenzyme in order to catalyze a particular reaction. So any questions on what you need to know for this slide?
You said the inactive enzyme and the active enzyme?
Yes, and not this stuff. Here are some examples of cofactors. They're inorganic metals and the enzymes that they work with. You guys don't need to know these. But cofactors are inorganic metal ions. Coenzymes are vitamin derivatives, and we're going to talk about many examples of that. So how cofactors work is here's an inactive enzyme here. A cofactor will bind to an active enzyme which facilitates a substrate binding as shown here. And that creates the correct environment for the enzyme to catalyze the reaction and produce a particular product. So we're going to talk about the two categories of vitamins. You have the water-soluble and fat-soluble. Water-soluble, we must consume those daily because they're excreted in our urine. We cannot store these in our body. Oh, also wanted to mention this. So one of my former colleagues went through this program, the PA program. We were in Germany stationed together. She came, completed it. She just took her pants like a few months ago, and she said that vitamins were on the pants. So this is probably one of the only things you guys will take from biochemistry that would be on the pants. All the other classes. A lot of stuff from all the other classes, but when she told me that, I said, "Oh, I'm gonna tell them. They need to be pants." But she did say that they were on the pants of some vitamins. So, as I mentioned, water-soluble vitamins, they are excreted daily in the urine. We cannot store them, and they are very labile. They're sensitive to light and heat, and they are precursors to a number of cofactors that are required to catalyze enzyme reactions. So we're going to talk about, this is kind of boring because I'm just listing the vitamins, but we're going to talk about the vitamins, water-soluble and fat-soluble vitamins, and we're going to talk about some of the hallmark characteristics that we see if we are deficient in the vitamins. So for example, vitamin B1 thiamine, I recommend knowing both names. This is associated with a coenzyme, thiamine pyrophosphate, and if we are deficient, we get very is a decrease in muscle function, a loss of sensation in our hands, and it can also cause cardiovascular effects. Riboflavin is associated with two coenzymes that we're going to talk about a lot because they're involved in the pathways that produce ATP, flavin adenine dinucleotide, and flavin mononucleotide. This vitamin can cause our urine to be bright yellow, and it can also cause skin issues such as dermatitis and a sore tongue. And here's the structure. You guys don't need to know that. Now, niacin, excess niacin can cause niacin flush, and this is skin reddening, skin itching, skin burning. Interestingly, a lack of niacin can also cause skin issues such as pellagra, and that is like skin inflammation and patchy skin darkening. Pantothenic acid, vitamin B5, it is associated with the coenzyme A. Coenzyme A is involved in the production of ATP as well. It's an intermediate in glucose metabolism and lipid metabolism. And if we are deficient in B5, we have cramps, and we can also have anemia. Peroxidine is associated with the coenzyme PLP that is involved in amino acid metabolism, and it can also cause dermatitis and anemia. There's a lot of overlap of these vitamins and cofactors because they're involved in similar or the same pathways that are involved with blood cell production, which is why you see anemia, and then growth, which is why you see retarded growth B7, this is one I take every day because I'm old and balding, so I knew what this one did. So, and it's also involved in depression as well. And vitamin B12, vitamin B9. This is very important in developing fetus. So if the mother is deficient in vitamin B9, folate, it can cause neural tube defects. We talked about cobalamin as being a treatment for cyanide, scavenging, and also arsenic as well as shown here. But it can also cause pernicious anemia, which is the lack of red blood cells in our blood. And then vitamin C deficiency is associated with decreased wound healing and connective tissue issues. And this was very common in the 15th and through 18th century because sailors wouldn't have exposure to fresh food, so they would have like their connective tissue would be very destroyed. Now the fat soluble vitamins. Easiest way to remember those, ADEK, fat soluble vitamins. These are not involved as coenzymes in enzyme reactions. They are lipid soluble, so they can potentially stay in the body and cause toxicity, but they rarely do. Vitamin A is involved in vision and RNA synthesis. It's an antioxidant. Vitamin D is synthesized when our skin is exposed to light, and it's, of course, we know vitamin D is very important in bone growth and bone health. Vitamin E is also an antioxidant, and a deficiency in vitamin C will cause us to have issues with our blood and potentially anemia. Vitamin K is very important in the blood clotting cascade pathway. There are several clotting factors that are vitamin K dependent, and so we need that in order to ensure we don't have prolonged bleeding and our bruise formation is occurring in a manner that will allow our bruises to heal properly. So, identify each of the following as water soluble or fat soluble. So what are the fat soluble vitamins? ADEK, so what's B9? C. B3. Right? Easy, right? Any questions? All right. So, what vitamin is responsible for collagen formation? Ascorbic acid. What about is part of coenzymes FAD and FMN? Say again. Riboflavin. Absorption of phosphorus and calcium in bone. D, right? Involved in vision and involved in blood clotting. All right. Any questions? All right, and I created this table for you guys. Are there any questions? Yes.
So just for, especially the fat soluble vitamins, is knowing the vitamin D sufficient, or do we need to know the full name of it?
Know the full name for all of the vitamins, fat soluble and water soluble. And I have this table. Any questions? All right. I will see you guys later on. Good luck.